Hello, everyone. I am very pleased to welcome you to the inaugural seminar of the Biosecurity Game Changers Fellows Seminar Series, coordinated by Tokozani Nyasulu and myself. My name is Sandra Matinye, and I'm honored to be one of the Biosecurity Game Changers Fellows. Today's topic is early warning systems, disease detection, and wastewater biosurveillance. In today's seminar, we will be exploring the critical role of early warning systems, disease detection, and wastewater biosurveillance in identifying and mitigating emerging biological threats. By examining these interconnected areas, we aim to enhance our understanding of how proactive monitoring and innovative surveillance methods can strengthen global biosecurity and public health responses. I will now pass it on to our moderator, Edyth Harper, one of the Biosecurity Game Changers Fellows, to moderate the seminar. Over to you, Edyth.
Thank you so much, Sandra. As Sandra said, I'm Edyth Parker. I'm also a Game Changer Fellow with the Brown Pandemic Center, but I am also a genomic epidemiologist with the Institute of Genomics and Global Health, or IGH, at Redeemers University, that is led by Christian Happy, which is why it gives me so much pleasure to introduce our first speaker today, Professor Christian Happy, which is a leading force in genomics, not just on the continent, but in the world. But he empowers the African continent through research. After training at the University of Ibadan and Harvard, he founded the Africa Center for Excellence in the Genomics of Infectious Disease, or ACEGID, in Nigeria in 2013. And almost immediately, ACEGID's rapid response and genomic analysis during the 2014 Ebola was crucial for containment. Since then, as director, Professor Happy has trained more than 2,000 African scientists, led groundbreaking work, including the first SARS-CoV-2 sequencing on the African continent, the development of a CRISPR-based Lassa fever diagnostic, and most recently, the sequencing of the first whole human genome on Nigerian soil as ACEGID transitions to the Institute of Genomics and Global Health. So there's really no one better to tell us about early warning systems, especially in the Global South, than Professor Christian Happy. Welcome. Prof, you're just on mute.
Thank you, Edyth. Thank you for the introduction. Thanks, I appreciate it and glad to be here. So I think I will be giving you a bit of a background and then to tell you how early warning system is very important for pandemic preemption or pandemic or epidemic response. Edyth mentioned 2014, and then that was a landmark year for us to demonstrate how innovation can drive epidemic response and actually introduce the concept of early warning when it comes to epidemic response. Not that prior to 2014, anytime there was an epidemic in Africa, there was a system set up by the World Health Organization to respond. Experts in quote were often sent to Africa. They would come and work and then tell folks on the continent what to do. Samples were taken over to to specialized lab in Europe or to some labs elsewhere in the world. Analysis were done, and 2 years later, they will tell us or tell people, in the countries concerned, what was the cause of the epidemic. However, having established the African Center of Excellence for Genomics of Infectious Disease with funding from the World Bank and US NIH seed funding, we really were thinking about responding to epidemics in a very different ways. We really had a concept of disease surveillance using genomics when genomics was not even thought about being deployed on the African continent. But we're very daring in our views and very daring in our mind and daring in our approaches and change the narrative. Just like your course is a game changer, we really thought about changing the game, and we did. And this is how we did, and then actually how early warning becomes important. When the Ebola outbreak started, in the rural confinement of Guinea in 2014, We had a premonition that it was going to spread out of Guinea, but then the question was, if this spread, what were we going to do that was going to be very different from the way epidemic responses have been done in the past? We had harnessed the technology and the tools of genomics, and we decided to prepare. And how was our preparation? You know, one thing is very important for this, epidemic response. It is, first of all, to understand, the pathogen, or having the ability to detect the pathogen that is causing the disease. And that we know clearly that if you cannot detect the pathogen, you can control it. If you cannot diagnose the disease, you can control it. We realized that for the case of Ebola that we needed to equip labs in West Africa, with the ability to diagnose disease. And we went on quickly to actually set up the very first molecular diagnostics for Ebola, and then we trained people in Sierra Leone, Nigeria, Senegal, and other West African countries. And as we had the premonition, within a few weeks of training folks in Sierra Leone, we had a spillover case. We have a case that moved from Guinea to Sierra Leone, and then one of our scientists, Augustine Goba, for instance, here, detected the first case. But then we also activated our own concept of what we call contact tracing, because we were doing work on Lassa fever prior to that. Within the first week, we were able to identify 35 cases, confirm 14, and then gave the data, share the data in real time with the government of Sierra Leone. But what is important here is the fact that, as we actually shared those data, we also went on to sequence those data. And that was for the first time during an epidemic where we were actually introducing the concept of real-time genomic sequencing, using the data actually to help epidemic response. But in addition to just doing real-time genomic sequencing, we did another thing that was unprecedented, which was basically just creating what we call open data access, open data sharing, because we made the sequencing data that we created openly accessible to the global health community for use to create vaccines, diagnostics, and other countermeasures development. But importantly was the fact that, we also realized that it was important to build a workforce. As we're doing all this work, we're training and building the capacity of folks in the region on how to not test, but also to do sequencing. Thirdly, we observed around June 16, 2014, to be precise, that one of the variants, based on our sequencing, has taken over. And really has taken over the 3 or 4 variant that we observed. So the question at the time was, what is making this variant, spreading much more faster than others? And we went on actually to characterize it and then to do some functional work. And then we established the fact that it has a lot of mutation, among which 27 were fixated. And then those mutations were very much involved, in the rapid invasion of the virus into the cells. That was a discovery of the concept of variants of concern. Fourthly, we realized that, our ability to use just PCR as a way to diagnose the disease was a big challenge because of the number of samples, and that created a lot of backlog, and then that really was an issue. Because most patients presented with fever, we noticed that some patients, even with malaria, were going to the Ebola triage center, got infected there, and then taking Ebola back home. And we basically realized that the main thing to change the game and then to flatten the curve or bend the curve of this epidemic was absolutely to create a test that can help you to have the result, the testing, the diagnostics in real time or near real time, so that at least you could actually report the cases as soon as they come. We went on to introduce a concept of translational genomics during an epidemic. So that is basically going from genomic sequencing work, and within a few months, we were able to create a very first rapid diagnostic test, 10-minute test that was approved by the WHO and then the USFDA under emergency use authorization. Bringing all the ingredients that I've just given you now actually created, the framework that the world used in 2020 to respond to COVID-19. First, near real-time genomic epidemiology, data sharing, translational genomics. You could see that we could go from genomic sequence data to vaccines in 9 months, because we had demonstrated that you could go from genome sequences to vaccines. Thirdly, also building the capacity and then training people on how to use all those, and then the discovery of variants of concern. So this is how we really introduce a game changer. I will change the game for epidemic response by bringing all of that framework that the world used for 2020. Fast forward actually to show how impactful our approach was, is the fact that, you know, a few months after that, we had a very first suspected case in Nigeria for Ebola in 2014. And guess what? We set up all this system and this machinery in Nigeria. Within hours of seeing the suspected case, we had the result. And within 2 days, we had the sequence. So, and that really put Nigeria on a different pedestal when it came to responding to that outbreak. A country of 230 million, with a virus appearing in a city of 25 million people like Lagos, Nigeria being the busiest airport, has been the country with the busiest airport in Africa and then the busiest seaport. There was a huge potential for the virus to spread from there to the rest of Africa and create a major, what I call, health calamity. But we did something that was unprecedented, activating the system that I've just described to you, and eventually work with government authorities and then contain this outbreak in an unprecedented way. In 93 days, we contained an Ebola outbreak with only 20 cases and 8 dead. That shows really the firepower behind the approach and the technology that and our approach to epidemic response. And that really demonstrates, you know, how you could use this to respond to an epidemics. But one thing that characterizes absolutely what I'm describing here are two things. The things that are characterizing all I've described here are two. One is speed. Second is accuracy. We had the speed to respond. In the case of Nigeria, we also had the accuracy of the data, because as we're generating those data, we're very accurate into the way we're sequencing, we're generating the data. We could use that as a way to respond in a way that other people couldn't. That is really the way we changed the game. And then we showed that we could do it here in Africa, show people how to do it in Africa, and then how we can actually also, you know, contribute our own quota to global, you know, epidemic response. And I do remember when the US had their cases, the first place where they sent their expert was in our lab to try to come understudy and understand the way we're doing it. So fast forward to just to show you that, you know, clearly when you do such things, then you're not just, you know, doing this to respond to epidemic, but you can also use the same approach to respond or to prepare the world for future epidemics. And then we use the same thing approach, for instance, this is like Lassa fever, sequence, you know, full genome, provided the first genomes of the Lassa fever virus, discovered the ancient origins of the virus, but then with those data, with the viral sequences, we accelerated, you know, the wave, vaccine development. And then I can present other cases whereby in November 2018, you know, 279 children died in the southern part of Nigeria. They didn't know it was a mysterious disease, in quote. 22 samples were sent in our lab, for instance, and then we were able to establish that it was yellow fever, you know, of lineage of variant that was circulating in the country that have not been seen in the past 94 years. So this is basically just showing you how you could apply this. And then the last one we did, as Edyth mentioned, you know, developing or sequencing the virus for SARS-CoV-2 in real time, sharing the protocol with the rest of Africa. And you could see Africa taking the lead in discovering all the beta and then the Omicron variant. And then lastly, for instance, is how we're able to use, again, the same approach to respond to the Marburg epidemic in Rwanda. You know, we came out of that with the lowest case fatality rate for a Marburg outbreak, characterize the virus in real time, use that as a way to respond to help epidemic response in country. An outbreak that happens in a major city, like Kigali, for instance, but you just end up with those few, with that few case fatality rate. That's definitely show you the firepower of bringing together genomics, epidemiology, bioinformatics, and all of these other tools and approaches, you know, toward, you know, early warning system. And that system that I'm referring to is what eventually we call what they call the Sentinel, you know, program. We've called it now the Sentinel program because it's an early warning system for pandemic preemption and response. Thank you.
Thank you so much for sharing your wealth of experience, Prof. We'll get back to the Sentinel project and some of those concepts in our panel discussion. But we'll move on to Dr. Joshua Levy, who is a Scripps research project scientist who develops computational tools for pathogen surveillance. So after earning his PhD in applied mathematics from Northwestern, he joined Christian Andersen's lab at Scripps, where he led the development of Freya, which is a widely adopted tool. And when I say widely, I mean more than 200,000 downloads. But the tool is basically for analyzing pathogen lineages in wastewater. And this tool is used from everyone from the CDC to kind of private labs. But he, of course, leads and contributes to numerous international collaborations on wastewater surveillance, including the Mujaji project with the NICD in South Africa. And he serves on multiple working groups for the Public Health Alliance for the Genomic Epidemiology, or PHAGE. And this includes the wastewater surveillance group, as well as standards development. So again, there's no one better to tell us about wastewater surveillance than Dr. Joshua Levy. Welcome.
Thank you. I can't currently start my video, but I think one of the hosts should be able to enable that. And thank you very much for the kind intro, Edyth. Good to be here. Thanks for having me.
Maybe while the, okay, are we
there we
seeing
go.
Josh? Okay,
All
there we
right.
go.
Great. All right. I'll hop into a brief little presentation, talk a little bit about wastewater surveillance. All right. So hopefully you should all be able to see that. Thanks again, Edyth. Hop into some wastewater related surveillance, and how that's sort of expanding the sort of purview of pathogen surveillance, and how we can really track pathogens in real time using passive but still actionable surveillance. So when we actually think about tracking cases of an expanding outbreak, we can think about sort of an evolving evolutionary tree of individual cases that stem from some likely index case. And many of these, especially when we're thinking about asymptomatic cases, cases that might not be ascertained for any of a number of reasons, we're going to miss a lot. So in the early stages, we're probably only going to be testing, potentially sequencing a small fraction of these cases, and many of them are going to be totally unobserved. And using methods from genomic epidemiology, phylogenetics, we can infer some of these points, but quite a lot of this sort of evolutionary structure and general population dynamics are really going to be absent from our ability to track viruses. So as an outbreak is going, and sort of expanding number of cases are slowly ticking up, and we're trying to figure out, is this going to be a major issue? Is there a new variant that might be spreading in the population? One of the key tools that we have to do this, and as Prof. Happy was just speaking about, is the ability to sequence, to be able to differentiate. Is this flu different from another flu that we might have seen before? Is this a different SARS-2 variant that might be expanding and cause a new wave? And our ability to sequence those in a large fraction of cases that do test positive, and doing so in sort of an expedient manner, is key to being able to actually find these variants before they're well over, a point where they can't necessarily be mitigated easily using possible non-pharmaceutical interventions. And one major issue here is that although we've heard a lot about sequencing and variant identification for SARS-CoV-2, when we look at other pathogens across the world, very few sequences are actually generated for many other pathogens. And this problem is even more an issue across the world as stratified by income. So low and middle income countries, we're seeing much less ability to track a lot of the pathogens circulating, and of efforts, exactly as Prof. Happy was speaking about before, are so key to expanding not only these sorts of health inequities that may exist, but also just in general, flu being one of the most sequenced pathogens, our ability to be able to track and monitor these pathogens in the population are so dependent on the development of networks like the Sentinel network that Professor Happy was speaking about before. So one tool that we've looked towards to be able to possibly supplement and provide additional information about pathogen spread is wastewater. And here, this is a tracking of both cases and wastewater viral loads here in San Diego, where I'm based. And you can see quite closely, there's a strong correlation between case counts and viral loads in wastewater, and this allows us to be able to track in advance possible increases in the population directly via wastewater. So this was sort of the initial phase of looking at wastewater, but it turns out that wastewater is much more powerful than perhaps we originally realized. Not only is wastewater much easier to do than running a traditional clinical collection approach, it's also very cost efficient, requires minimal changes to lab workflows, and is perhaps most importantly, very representative of what's happening in the community. You're not necessarily reliant on individual people getting tested or bringing samples from place to place in order to be able to figure out whether or not you might have an issue in your community. And as Edyth mentioned, this tool that we've developed called Freya allows us to track pathogen lineage dynamics directly through sequencing of wastewater. So to get a sense of what this looks like from the Omicron perspective, here in San Diego, we're able to identify in advance the appearance of Omicron in San Diego about 2 weeks ahead of detection via clinical sequencing. And we can also check to see the overall contribution of each of these variants in terms of its importance in driving possible waves. And this is not something that we can do just, in San Diego, where, we have the wastewater treatment network that I just showed a little map of. This is something that we're doing everywhere in the world, but using different strategies as needed for each individual place. So this is part of one of our recent projects working in Blantyre in Malawi, and we're able to get pretty much the exact sorts of results that we get in San Diego here, just by taking into account things like population density and flow of possible rivers and streams in the community to be able to identify key sites that we're able to monitor. And this is just looking at rivers and streams and canals, etc. So what we can see here is that in very much the same way we see in San Diego, we're able to track waves in terms of positive detections at these different sites and rivers and streams, and we're able to sequence that wastewater and environmental samples in order to actually track these dynamics over time. And we're really doing this all over the place, including in Nigeria, at ACEGID, now IGH, with collaborators in South Africa, in Malawi, as I mentioned, in India and in Bangladesh, for example. And it's these sorts of same kind of conserved approaches that allow us to really rapidly spin up these systems, strengthen these systems across the world, and start looking into sort of a unified structure for pathogen surveillance. And the way that we think about this is we're sort of taking all of these different dimensions, starting with clinical surveillance and actually doing basic testing, to wastewater surveillance and quantification of levels in different places, bringing those pictures together, and then bringing in the genomic side of this equation. Can we actually sequence these samples, figure out which lineages are actually causing these particular outbreaks, see how that is mirrored within the wastewater, and then have this sort of integrated picture of what's happening clinically, what's happening in wastewater, and allow them to sort of provide information about what we might be missing in one that we're seeing in the other, and vice versa. And this is really the sort of key picture that I'm really pursuing in a lot of my work. So we're trying to build a lot of the core structures in order to pull this information from publicly shared data, so shared with sequencing data that's available on shared repositories, as well as global databases like the WHO, to be able to track epidemiological signals. And we want to bring this sort of picture into a single sort of conserved or sort of integrated picture and make this available to as many people as possible. And one of the main efforts that we're using to do this is our outbreak.info method, which integrates information across all of these distinct sources to get a sort of single picture of what might be happening in the community and make that available to public health stakeholders as well as just general members of the population. And to see what this looks like in California, for example, you can see that we're able to track not only clinical and wastewater signatures simultaneously, we're able to also look into the sequencing data and see how these signals may or may not match to one another. And this is something that we're thinking not just, again, in terms of the US, where we have the National Wastewater Surveillance System with lots of data coming in for sequencing of wastewater. This is something we're thinking about in a very global perspective and for many pathogens as well. So we're not just thinking about SARS-CoV-2, we're thinking about measles, mpox, flu, etc. And we're developing the sequencing schemes to be done at low cost that are pathogen specific, whether that's focused on individual SNPs and resistance markers to entire genomes, depending on the pathogen. And we're building all of the actual bioinformatic tools to make that possible from the analysis perspective, and the visualization tools to be able to actually handle this data, show it back to the population and say, actually, this is what's happening in your community right now, and allow people to make judgments as to their own health behaviors and to inform public health stakeholders. And we're doing a lot of this within the context of phage, is a group that I work closely with, especially thinking about wastewater standards development. We're developing all sorts of standards for how do we think about site selection, sample collection, lab processing, etc., to be able to provide these sorts of standards. And not directly mentioned in the main part of the slide is actually ethical and legal concerns. So thinking about when we are sharing data, how do we share it? How do we make sure that it is providing, actual benefits to the community, minimizing harms? These sorts of things are really important towards the pursuit of this research. And it's really what allows us to extend into these really global networks, to really bring all of these different methods, all of these different teams and researchers across the world into a unified sort of network of pathogen surveillance. And just to see what this looks like, if we can bring in both wastewater and clinical sequencing data to be able to track pathogens in near real time. So this is sort of integrating all of these efforts across standards development and centralized dashboards, all into this one picture of pathogen surveillance. So with that, I just have a lot of folks to thank, but I'll not name them and just put this slide up for the moment. Thank you.
Thank you so much for those very key insights born from extensive experience, Dr. Levy, and for emphasizing the importance of open science and open data for actionable information in outbreak analyses, which is something I'm sure we'll get to in the moderated discussion. But we'll move on to our next speaker, which it gives me great pleasure to introduce Dr. Marta Giovanetti, who is an assistant professor at, excuse my pronunciation, Università Campus Biomedica di Roma in Italy. And she's also a researcher at the Oswald Cruz Foundation, or Fiocruz in Brazil. She earned her PhD in molecular evolution of RNA viruses from the University of Rome, but has conducted field work on numerous viruses, including Ebola in Sierra Leone and arboviruses in South America. Her research uses phylogenetics, which is some of the stuff Josh was touching on, and phylogeography to study viral outbreaks, integrating, like Prof and Josh has been talking about, these different data streams, including genomic data, but also spatial and ecological data to inform public health policy, particularly regarding climate change's impact on mosquito-borne disease like Zika, dengue, yellow fever, and most recently Oropouche virus in South America. So once again, there's no one better to tell us about early warning surveillance systems, especially in the context of One Health and climate change, than Dr. Giovanetti. Marta, you're on mute.
Okay, got it. Thank you so much, Edyth, for the nice introduction, and of course, for having me today. And hi, everyone. So today I'm going to talk about, like the importance, and I'm going to share actually with you some of our results that we obtain in this, let's say, long history of try to track the emergence and reemergence of mosquito-borne pathogens in Latin America, actually. And in the importance of try to build up a kind of genomic surveillance effort all across the Americas region in order to try to understand even the driver behind their emergence and then expansion. So one of the main points is, of course, related to climate change and how climate change might be able to amplify disease and epidemics, right? And we know that actually in this topic, many things need to be considered. That's supposed to be, of course, the presence of ideal climatic conditions, the presence of competent vectors, the presence of course, human migration, the connectiveness of countries, and of course, like this kind of interaction within like human and their vectors. There are several of traits that we need to consider in the context of climate change that might be amplified by climate change, but my focus in this presentation will be like focus our attention on arbovirus with a specific on mosquito-borne pathogens. So this is a kind of top list of the one that's supposed to be the pathogens that might be amplified by climate change, and of course in this kind of list, the vector-borne diseases, they stand out as one of the main threat of national as well as international concern. So since a very long time, I mean, like almost a decade nowadays, we were actively involved in South America in trying to put in place a kind of genomic surveillance effort in order to try to understand this kind of very complex dynamic of different kind of viral pathogens which are like co-circulating in the country. I mean, like very recently, we try to extend this kind of activities, focusing not just on Brazil, but even like by working on behalf of the Pan American Health Organization, we got the chance to extend this kind of effort all across the Latin American region. So by recognizing also the importance and one of the main role of Brazil as national as well as international exporter of viral strains. So one of the main points is nowadays, like try to understand how we need to integrate different kind of data. They're supposed to be, of course, like not just the genomic data, but even like environmental data, evolutionary data, epidemiological one, but even like immunological one, in order to try to understand how different factors they might be able to act and potentially interact to model viral phylogenies, right? And how they might be able to intensify the transmission dynamics of some specific pathogen. But at the top point, there is like the necessity to try to obtain, of course, like whole genome sequence to inform public health response. That's supposed to be important to try to understand the origin of pathogen, try to understand how many introduction of a specific pathogen happen from abroad, then start to determine the presence of some specific community transmission, and even like try to detect the presence of specific lineages that's supposed to be associated with the emergence of a pathogen into a novel region. And then, of course, even try to identify some specific point mutation that's supposed to be associated with infection caused by this virus. So I would say that one of the main points of this kind of genomic surveillance effort is, of course, like try to determine and follow the real-time evolution of pathogen, but even like, you know, identify potential novel transmission routes, but even like try to identify the presence of specific point mutation that might be associated to differential clinical outcomes that might enable us to understand and provide information regarding the pathogenesis of those pathogens. And we start this kind of, let's say, activity during the emergence of a novel threat of international concern. By the time it was 2015, 2016 in Brazil, the Zika virus epidemic. So by working together with several collaborators from UK, we got the chance to implement by the time, for the first time, the use of the nanopore sequencing in Brazil. And since then, actually, and this, of course, this approach enabled us to describe and date back the introduction of Zika in the Americas. And then, of course, by using phylogeography, we were also able to determine and reconstruct the one that was the spatiotemporal dynamics. So one of the main points then was try to, I was then since then involved in the development of, let's say, a kind of novel approach for the study of infection diseases, and most with a major focus on on RNA viruses. So, and then we try, of course, to extend this kind of effort to other pathogens in the Americas, including West Nile, and then like dengue, chikungunya, and then we also got the chance to follow the emergence of yellow fever. So by the end of 2018, by the end of 2016, at the beginning of 2017, Brazil started to register one of the main outbreaks of yellow fever that was never reported before, that was able to achieve region that presented a very low vaccination coverage. So quite recently, let's say, we were able additionally to follow the the expansion of this outbreak in the south part of the country. And by the time we try to use and implement a kind of novel approach for the study infection diseases, in which we try to integrate a different kind of data, including, of course, the genomic side in a kind of One Health approach. So we got the chance to sequence the virus from human cases, non-human primates, but also from mosquito. And then we try to integrate this information with ecological, epidemiological data, including, from the ecological side, temperature, humidity, precipitation. So this gave us the chance to estimate the one that we used to call the index P, which described the suitability of the vector, which is responsible for the transmission of the virus. And we found a kind of synchronicity within the different epidemic peaks through time, and then, of course, the suitability of the virus. So this gave us the chance to understand that now we might be able to even predict potential novel epidemic peaks. And then we tried to use the Bayesian regression model in order to try to, pinpoint, let's say, some specific hotspot all across the southeast part of the south and the southeast part of the country, region that's supposed to be prioritized for active surveillance because of ideal, let's say, ecological and immunological conditions. So this work was, of course, absolutely important because by working very closely with policymakers, those regions were additionally were targeted for active vaccination campaign. And as I said before, through this kind of, effort and because of the establishment of this kind of toolkit all across the different Brazilian micro regions, we got the chance to understand and to identify this kind of cross-border transmission with other Latin American countries. And very recently, actually, just 1 year ago, we established this huge regional network. We are very proud of it. So the name is PAOGIN, and the PAOGIN has one main mission, which is try to bring all the different Latin American countries coming together and show that beside all the different challenges, they might be able to perform world-class science. So one of the main points, it was, of course, try to establish locally capacity building. I mean, in the context of genomic surveillance, generation of whole genome sequences, but also perform this kind of jump within data generation and data analysis, which is one of the big stepping points. Very recently, I mean, we additionally understood how not just climate change, but also this kind of active, extensive human activities might be able to amplify disease and epidemics. And this is why we start to perform this kind of, as we used to call, exercise. So we start to work and play a little bit with mathematical models in order to try to perform this kind of projection in the change in the vector population and suitability range. So we start quite recently to try to estimate this kind of drive and climate and environmental drive and transmission potential for mosquito-borne pathogens using this kind of eco-epidemiological approach. And this kind of approach gave us the chance to identify some potential blind spot all across the global south region that, because of ideal environmental and climatic conditions, supposed to be prioritized for active surveillance. And of course, this was quite timing also in the context of, this very recent reemergence of Oropus in Latin America and in Brazil. So for a very long time, this pathogen was restricted to the endemic Amazon region. But very recently, actually, the Brazilian Ministry of Health started to report an increase in the number of cases associated to Oropus infection. And these will start to be, those kind of cases start to be reported also in other Latin American countries. So including Bolivia, Colombia, Peru, and most recently also Cuba. And the CDC, by analyzing some travel-related cases, start to report also this case also in Italy, Spain, and Germany. So this just highlights that this kind of pathogen might be a kind of international concern one. So we strengthen this kind of surveillance at the country level. We start in Brazil, where we got the chance to describe the first fatal ever cases associated with the Oropus infection. And then we start also to elucidate some specific mother-to-child transmission route. So in this work, we try to just bring together many different central public health labs located in different Brazilian macro-regions. So we got the chance to describe for the first time this kind of rapid epidemic expansion behind the Amazon basin. And by using travel-related data, we were able also to describe for the first time this kind of long-distance movement from, the American regions to Europe. And very recently, actually, we then decide to try to understand the drivers behind this kind of expansion. So we use phylogeography in order to try to estimate the spatiotemporal dynamics of our push. So this kind of analysis gave us the chance to understand and to identify two specific phases, let's say. An initial phase during the time the virus was restricted to the Amazon region, and an expansion one during the time actually the virus was able to reach region behind the Amazon basin. So by analyzing the three different segments, we got the chance to identify this kind of long distance dispersal. And then we try to integrate different kind of data in order to try to detect the one that were, let's say, the likely drivers behind this expansion. So we found evidence that suggests that at least during the expansion phase, the virus was preferentially circulating in regions that presented a high human population density, ideal climatic condition, a lower evergreen broadleaf forest, and also a higher banana and cocoa cultivation, which also highlight how, of course, deforestation and this kind of extensive human activity have created a kind of ideal environment for the mosquito breeding, enhancing the chance of humans to be bitten by infected mosquitoes. So this enabled then the establishment of this kind of Global South Consortium, the climate. We are responsible for the Latin American side, let's say, which has as a main mission, like try to develop novel approaches for the study of infection diseases that might be amplified by climate change by focusing on ecology, epidemiology, and evolution. I just want to conclude to say that, of course, all of those activities, they were very linked with this concept of try to advance responsible research and innovation with education. So we were extensively involved in this kind of training course all across the Global South with a major focus on Latin America. So up to now, we got the chance to train more than 500 fellows from more than let's say 50 African and American countries together. So just few take-home message. I mean, like biosurveillance is, of course, the key to detect climate-driven diseases, policy engagement and through long-term investments, which is absolutely important. Of course, tech alone is enough, so funding and commitment matter more. And then, of course, the early warning must trigger rapid action, and of course, data integration improves outbreak prediction. Thank you.
Thank you so much, Martha. It's always extremely heartening to hear such good science, for me at least as a scientist, and also to finally learn how to pronounce the Oropouche virus, because I've been butchering that, it seems, for years. And so Martha has set us up incredibly well for our panel discussion, which we'll do with all our panel speakers in her final slide there, which is the first question I would like our panel to address, which is, and this is going to be a multifaceted and leading question. I'm allowed to do that. We're not in court. So firstly, what is your vision for the future of biosurveillance? I know a lot of you guys have touched on that, especially in the programs you've already developed. But what are the key characteristics of an ideal surveillance system, which is a question we got in the chat as well. But also, what technological advancements or scientific breakthroughs or data is needed to realize that vision? Or are the primary obstacles not technology, but rather sufficient funding of fundamental infrastructure and necessary political commitment? So we'll start off with Prof. Happy, and then feel free to reply.
Yeah, I think I'll go get started. I think the first thing that I will say, the threat, is keep funding. Funding is important. Without funding, we can't really do surveillance in a way that we can actually create what I call global security system. So, and I think I'm a bit concerned on the fact that, after every epidemic or every pandemic, we'll go back to business as usual, and then we get always cut back, as if nothing happened before, and then we'll keep scrambling anytime we have a major epidemic or major flare-up. So that's definitely one. Then secondly, I think there is a need actually to create an advanced technologies to a point whereby we can basically just, apart from doing modeling, to actually to be a bit very accurate in predicting, what epidemic could be like. There is really a need to, advance technology in a way that we are doing basically to characterize pathogen, to do surveillance in a way that we are doing holistically, using the One Health approach that has to do with human environment and animals in order to, to do surveillance. But then this has to be holistic. And when I mean holistic, it means that, as the samples are collected, we also need to do, as I said, there are two things that we'll probably need to prevent maybe the next pandemic. One is, the speed of execution, and then the third is accuracy with the data that we're creating. Then in doing that, we need to advance our sequencing platforms in a way that we can do it much more faster, we can do it maybe cheaper, and then the analytics behind it has to be also be done very, very fast. So, the platform that we have now are very good, but the thing is that it takes a long time for them to generate and create the data and to release the data. We need to have things that should be analyzing the data in a matter of minutes, so that we can actually have the data out in near real time. So I think these are some of the things, but they also ultimately, collaboration is very important. I think what we're seeing is very much a situation whereby we have a lot of groups, but that are working in silos. There's a need to bring everybody together. But in order to bring everybody together and in order to be effective, there's also a need to have policies that actually result in being really open. Really open means that, when people are reporting data, they are not stigmatized against, and then there is really no discrimination against. I think these are issues. Because if a country reports a new virus and then the people are stigmatized or discriminated against, it means that we are actually making backward steps, not forward steps. That is other thing that I want to point out there. But then also there is a need actually to set up a framework, not the kind of framework that we're seeing out there that will be done what I call that we have at WHO the last time. So I think what we need to do is a framework where there is absolutely equity, not a framework where some people are telling others what to do, but a framework where people agree to come together and work together toward the same cause to create a world that is more equitable, to create a world that is fair, where people can feel comfortable sharing the data, knowing fully well that there is a benefit for data sharing. There's no point encouraging, what you call global collaboration when the data are shared, and then at the end of the day, the data are taken, create countermeasures are developed, and then those that don't have the resources and who willingly share the data are not having access to the countermeasures developed. I think that is the legal framework that is really needed in order to move forward. Because I can tell you that, after COVID, we made a lot of steps. The COVID-19, we made a lot of steps backward because some people felt very, very used and utilized when it comes to data sharing. And I think there is a need to look into this responsibly and let people come together, and then do what I call a real pandemic treaty. Some of us wrote opinions in Nature actually to say, look, it's bullshit. Whatever we're talking about pandemic treaty, we need to be very open, and be transparent about the way we want to do this treaty. Otherwise, we'll be going back 50 or 100 years. Thank you.
Professor, you perfectly answered my second question as well, which is, and I'll throw this to Josh and Matt as well, together with question one, is, as Prof said, the emphasis should be on how we ensure equitability of access to the benefits from advanced surveillance technologies, especially in lower middle income countries. So I believe that answers the first question, because our vision is equity, I think everyone on this panel. But if you want to address the first one, the first question about what you think the future of biosurveillance should look like, and how do we build equity into that, as Prof has emphasized very correctly. Marta, if you want to start.
Of course. It's hard to talk right after Professor Epi, right? Because actually you already addressed almost all the point. So I would say that, at least from my experience in Latin America, but even in Europe is not that much different. I would say that one of the big points for this kind of biosurveillance is try to connect much more, like the animal health with the human one. Right, because they are connected, and I can say this because even, for example, in Brazil, so we have, of course, the Brazilian Ministry of Health, which is absolutely not connected with the one which is, the animal side. So this is one of the big points. But even like in Italy, for example, I can say we were very recently working on the H1N1 swine flu, and again, the Zooprophylactic Institute, they don't have any kind of connection with the human Italian Minister of Health, So I would say that the One Health approach is the key in order to, and this is mostly true, of course, for zoonotic pathogens. So one of the main points, it will be try to identify, this kind of spillover, but even spillback events. Another very important point is like try to develop novel approaches that might enable our chance to respond to epidemic, not just during the post-epidemic phase, you know. So we might be able to predict potential novel epidemic peaks and even novel, I mean, like the emergence of novel threats. And I think that maybe Josh can say something more on this topic because of this amazing toolkit and implementation of wastewater surveillance as a very nice early warning system. So I can say that, for example, in Europe, in Portugal, was used this kind of toolkit in order to try to predict the emergence of mosquito-borne pathogens. I mean, like, need to be extended in a kind of global scale. And the last point, again, at least in my opinion, is like try to extend the capacity building that all the different countries acquired during the pandemic in order to make them able not just to obtain whole genome sequences, which nowadays is quite easy, right? But even like try to enable them to analyze their own data in a kind of real-time way. So, yeah, that is.
Josh, now you face the challenge of speaking after Dr. Giovanetti, and it just gets worse
after
down the line.
both of them is tough. But that's very kind of you to say, Marta. I think my sort of way of thinking about sort of the future of a lot of this biosurveillance is these integrated networks, whether that's more One Health, animal-focused spillover sort of monitoring, thinking about passive surveillance networks, which is a major focus for me, thinking about how we can, at low cost, have consistent surveillance of key pathogens of public health interest, and be able to respond and scale up possible active surveillance networks, active responses, and public health guidance and interventions in response to that. If we have all of these sorts of pieces, we're kind of covering all of the key dimensions. And as Prof. Happy and Marta both already discussed, one of the most important parts to doing that is making these technologies open source, publicly available, and affordable. I think one of the key things that wastewater surveillance brings that I like so much about it is that it allows us for a very small cost. You know, we're talking on order of tens of dollars per day to monitor an entire city in one go. So we can be just tracking an assortment of different pathogens quite sensitively directly via the wastewater. And then we can get, you know, if we see something that's, you know, a One Health concern or a possible outbreak in a human population, these sorts of things where we can contact the appropriate people and start proceeding with those active surveillance networks. So I think that's sort of a key thing. But none of this happens unless we can build the technologies and distribute them and train people to do this work kind of in a global scenario.
Yeah.
Sort of fundamental.
System strengthening, I think, is a vision we can all get behind. But seeing as we have a very lovely mixed audience today of, you know, scientists and people on the policy side, I thought I would ask you three, as, you know, public health scientists, as well as people who've kind of delved into, you know, the interface with policy, about how can we actually talk to our policymakers and demonstrate, you know, the long-term importance and cost-effectiveness of investing in surveillance and kind of these reactive response systems, particularly in remote or resource-limited communities. How do we go about talking to our policymakers about that? And if we can keep it short, there's a few questions in the chat from the audience we'd like to get to. But Prof. Happy, as someone who very frequently speaks to policymakers, can you give us a few tips and tricks on how to?
Yeah, I think one of the things is actually to have data and then have use case. And then once you have the data and then you make the data actionable, the policymaker wants to see how your data can actually translate into, you know, tangent evidence. And that's really what we have to do. You know, we don't have to keep the data. We need to make sure that we share the data, you know, with them. And then as we make the data actionable, they can see actually the impact of your data in the intervention. So, I mean, I mentioned, for instance, the case of the 279 children that are in Nigeria. They don't know what it is. And then once we use a metagenomic approach, find that it's yellow fever, share the data with them in real time, then they can go and actually just use the data actually to make the intervention. And I can tell you they did that, and within 2 weeks, they contained that outbreak, saving thousands of lives. That kind of action makes a policymaker say, look, this whole, genomic or whatever that this guy is doing has direct impact. I can see that. I think we need to do as scientists, share the data as we have them, and then also work with them in order to implement the policies, than to first of all think about making papers. Again, I think we need to forget about science first. Let's do science for the people than to do science for the papers.
I think that's perfect, and I know it's an ethos we share, is that we're all public health scientists first, right? Rather than we run programs instead of papers, right? So Marta, I know that's something close to your heart as well. Do you have something to build on there?
Yeah, well, as I used to say all the time, it's not just about like executing a kind of protocol, but it will be like try to adapt the methodology that we are proposing with the local needs. So, and of course, show that the methodology that we are proposing is not just linked to a research article, of course, but is linked to, try to implement kind of try to track actually the emergence and the impact of novel pathogens. So of course, it's not that much easy, and you need to try to build up a kind of, nice connection with, of course, policymaker, but then, as I said before, it will be not just execute a kind of methodology, it will be like all the time, try to understand the local needs and try to adapt what you are proposing to what is happening in a specific place.
Yeah, that's perfect. Context is key, right? And I know this is something Josh thinks about quite a bit as well.
Yeah, I certainly think that being there to help build some of these systems is a key aspect. But one main focus of the work that I do is trying to make myself kind of redundant in the end, is to build the technologies, train the researchers, and then let things sort of go from there. I think that's sort of my aspiration here. And in terms of whether data is held for a paper, that's not really a focus of mine. But what it is and very important for is this can have serious political ramifications. Both Marta and Prof. Happy already talked about how the sort of control and the sovereignty of data is really a key issue. And whether that means that, data generators have total control over how this is published or discussed in the media, or whether it's published at all, that's not my prerogative. I want this data to be available to public health stakeholders, to members of the public that need this information, but it shouldn't be used, and we have to be very careful. And it's a major focus of what we do in the phage ethics working groups, and as well as in our wastewater working group, is figuring out how to take this information and actually use it in a way that it is productive from a public health standpoint without causing any undue harms to the population that we're monitoring.
Big questions you guys deal with, and I'm sure very, very long meetings. But we'll just have one quick question. There were so many great questions in the Q&A, and I see Dr. Levy has been active in responding to them. But we'll just answer one, and this one is pitched to Prof. Happy, which is, you cited that the speed at which you acted and the accuracy of the data collected was a game changer in your successful arrest of the outbreaks in West Africa. So this is, the Ebola to start with. But the question is, what are some of the key aspects of your preparation that enabled you to respond so rapidly? So what is the work you do to prepare?
What you don't have to, you have to prepare when you don't have an outbreak. That requires for you to build and training. Training and capacity building is very important. If you don't have those, if you don't train and build the capacity of people, set up the infrastructure, and then make sure that you also are ready in terms of technology, then actually you cannot respond. I think that's all I can say about that. So development has to happen, it's a continuous process and happen in periods where you have nothing but continuous training, because the people are the hands that you need to have on the field.
Again, it comes down to system strength, and we could have a full conversation, especially as there's a lot of early and mid-career people on audience, about what skills we need, how do we go about get that. But unfortunately, we've run out of time for today. Unless Wilmot, I saw your hand was up. Was it a very short question?
No, I just wanted to make a quick point. So I'm building with a team and modernizing South Africa's surveillance system, and working together with the National Institute for Communicable Diseases and with CLIMAID, with Tulio D'Oliveira. So I just wanted to mention that on a policy. I'm a policymaker. I'm a former member of parliament, so from South Africa, and we're working together with the Minister of Health, the Minister of Agriculture, and the Minister of Environmental Affairs. Those are 3 data streams: environmental surveillance and genomic surveillance, which sit in, it's run through the National Institute for Communicable Diseases, but the ministerial responsibility sits across ministries. So there's a need to integrate those systems, and we're working on doing that, getting presidential authorization. So, and to say that just 2 points around wastewater surveillance is the challenge of identifying proper signals in the middle of all the noise that comes through, and then, you know, what are you asking the policymakers to do exactly? So in some cases that's clear, in other cases not clear. So that's something that needs to be refined. A second point around Joshua, I'm afraid you can't quite do that, right? So there's a real challenge right now, especially with improving technology in the area for continuous education in bioinformatics, for example. So the capacity building side and upgrading side is a continuous effort. So that infrastructure has to be in place before you sort of go in and out, okay? But just to say, this was great. I listened in to most of what happened. I'm in Seattle right now, and at the airport. So, but I was delighted to be part of this. I'm sorry I was a bit long-winded, but to say this is great.
Oh no, that's fine. I think Josh meant he wants to create independence in the local context rather than hit and run. Yeah, as someone who's worked extensively with him, he's absolutely fantastic taking scientists and keeping it sustainable as well. But I see we're almost at time. Well, we are at time. So I was going to pose one question to our panel, but instead I'll pose it to our audience to take away with, and maybe you guys can all message the Brown Pandemic Center Blue Sky account and tell us your answer to this question, which is, what is one thing about biosurveillance that doesn't keep you up at night? Because there's a lot of anxieties around, you know, H5N1, Marburg now in Tanzania, and there's a lot of doom and gloom. But what is one thing, what is one development or trend that gives you hope for the future? So just as a takeaway there, we can get our thoughts from our panelists, maybe over on Blue Sky, or Prof. Hoppe, if you want to jump in quickly, maybe under a minute.
Yeah,
unless you want to say the future.
Yeah, I would just think that the future, I'm very hopeful. Like, I think what I've seen over the years is that the next generation, or what I call the Gen Z, they've broken down the barriers and then the silos. They've broken down what the politicians have built, and then they are very, very much, I mean, way more open to work together in a very collaborative ways. They are less nationalistic than what, I mean, the generation of politicians that we have in power these days. They are very much more, they are more global in their thinking. And I believe that, you know, in the long term, these are the people that will set up a pandemic treaty that will be equitable and fair. And then where I think that generation will create a world that is better and a world that is more equitable. I think I give the Gen Z, for instance, that, I mean, I think the hope of the world is in their hands.
As a millennial, I'm only slightly insulted, Prof, but I'll include myself in that bucket for now.
Some millennials are in position of power, and I can tell you that's the reason why I'm excluding that whole, including my own generation.
Yeah, this is millennials, as we call ourselves. But I'm only left with asking you guys again, what doesn't keep you up at night? What gives you hope? And hopefully, you know, let's live our lives to prove Prof Hoppe right. And see you guys maybe on Blue Sky with your answers. And if not, thank you guys so much for your time today. Thank you so much to our brilliant speakers. you can read their papers if you have any questions left about their brilliant work, or contact them directly. And if not, a big thanks to our seminar conveners, Sandra and Thokozani, for their wonderful leadership, and the Brown Pandemic Center for facilitating this wonderful fellowship and conversation. Thank you guys so much.